Hi, Akash. How are you? Can you hear me? So the unmute button is all the way on the bottom right. There's a little microphone symbol. If you press on that, you should be able to unmute. If not, I have to restart the room because since you started the room, you're moderator now. So I cannot put the slides up or anything. So let me just restart the room. I'm sorry. Or okay. Hi everyone. We will start on top of the hour. So, hi, Ashka.
Hello.
Yes, now it works perfect. Yeah, the first
Okay,
time you
perfect.
use the app, I think I don't know why they do it for security or
All right.
something, that you
No.
cannot unmute the very first time. So now it
Sure,
works.
but it's working now, correct?
Yeah,
Okay,
perfect.
perfect.
Let me put up the topics and then the presentation. Okay. And then
So
I'll show
just
you.
to, sorry,
Oh yeah, go ahead.
just a quick question since I'm not too familiar with this. So um, is this a very general audience?
Yes. Yeah, it's
Okay,
a mix of our audience, exactly.
okay,
So,
okay. All right. So,
yeah, it's a public room, so anyone can
okay,
just join in general. So it's good to have like a mix of like the general view and then also
hmm
details for the science
okay.
people. Yeah. Thank you for coming and doing this.
Sure, it sounded
On a
interesting.
oh great. On a Friday evening for you.
That's quite all right.
Yeah. So yeah, we have like around 7 minutes until we start. I'll
hmm
share in the meantime on Twitter that we started, and then before we go into your presentation, I'll introduce you, and if it's okay with you, I'll ask me a couple of interview questions, like around how you became to be a scientist type of questions, if that's okay.
That's fine.
And
Yeah,
yeah, I think for like the general public, it's very
sure,
interesting. So
sure.
perfect. Hi everyone. I'll put the slides on top, and I will be posting right now in the chat the open source version of the paper, which is a bioarchive preprint, and then I'll also add the Nature paper version. So you have the option to read both, but in case you don't have access, there's always the preprint. And the slides are pinned on top of the room so everyone can access it. And yeah, feel free to share the room. I'm going to do that right now. Thank you. Okay, I shared the room. We still have a few minutes, but I hope your year started well so far.
Sorry. Yes,
It's
just
been
kind
good. Oh yeah, don't worry about it. It's just.
I'm sorry. I may have missed it. Did you say something?
Oh yeah, I just said I hope your year started well,
Oh, yeah, yeah.
better than last year,
Yeah.
hopefully.
Yes, for sure. How about you?
That's great.
Yeah.
We had some people dying. This
Oh,
holiday season, which was really sad.
yeah.
But other than that, everything is good. But I read that statistically, it's much likelier for a person to die in December for whatever reason, even in Australia where there's
Yeah, I was
summer.
going to ask. This was the first thing that came to my mind. Is it even in Australia?
Yeah, that was curious. I don't know.
Hmm.
It's interesting. Maybe just lack of
So,
sleep and drinking. I don't know.
yeah, like some cultural thing probably, rather than weather related.
But other than that, everything is good.
Mhm.
So, hi everyone, welcome. We will start in a couple of minutes. Feel free to check out the paper in the meantime. There's a preprint version which is free for everyone in case you don't have access. And yeah, I'm looking forward to this really interesting discussion. Um, it's a really interesting study, and also when I read your bio, the research you worked on before was really interesting. So I'm really looking forward to this. So.
Yeah, thank you. I'm very much looking forward to it too.
Okay, just a couple of minutes left. I think people will still keep coming in. Like usually, people keep coming in a few minutes also after. I think we can just start on time and then we'll go from there.
Okay.
So yeah, welcome everyone to Science Society, and of course a special welcome to you, Ashkash. And as I said before we start, I'll give the audience like a brief overview so they get to know you a little bit. Um, and let me really quick share your lab website into the chat so people the cool thing about recording here on Clubhouse is that the chat and all the links we share, also the slide link on top, they'll stay active. So when people listen to the replay, they can go into the chat and click on those links if they want to take a deeper dive into anything we discuss or any of your work. So okay, so Dr. Ashkash Basu, he began his academic career at Stanford University in the group of Zeph Bryan as a PhD student. And he there was visualizing biochemical reactions, basically one molecule at a time, using biophysics tools. And then later on, by the end of his PhD, he was awarded with a Life Science Research Foundation postdoctoral fellowship. And that's when he joined the group of Jim Hudspeth at the Rockefeller University. And there he developed a magnetic tweezing based system, what was capable of interrogating the entire inner ear tissue, which is also really cool. And then, as later on, he joined the lab of Teiki Pa at Johns Hopkins University, where he was a postdoc. And he continued working on single molecule high throughput assays for studying DNA and chromatin at the genomic scale. And that's where he built the expertise in using single molecule fluorescence resonance energy transfer, which is a really cool tool. I use FRET in my work, but not the single molecule level. And it's a really helpful and powerful tool. And then in 2022, he joined Durham University as an assistant professor. And we will talk about his current research now during the talk. But before we go into your presentation, um, what inspired you to become a scientist? If you can recall, maybe you always wanted to do that as a kid. Maybe it was a book or a teacher that inspired you. Whatever the story is, we are really curious to hear it. Thank you.
Um, yeah, that's a very difficult question to answer properly. But as far as I can recall, I have wanted to be a scientist since I was a kid. I mean, it's goes back as far as I can recall, as I mentioned. Although my specific scientific interests were, of course, always shaped by information and resources that were available to me at various stages in my life. Um, as a kid, you're impressionable, and you tend to get more interested in the sort of things that you hear others talking about, or the sort of literature that you find available. And if you look at popular science, which I would say was certainly instrumental in getting me interested in science, there is a lot of popular science related to topics like astronomy, astrophysics, cosmology, and all that. And so that's what got me interested. But then later, when I went through the process of science, when I went to university, um, I got more interested in biological problems and biophysics and life in general. And that's how I switched over from a more physics a sort of direction to the biophysics research that I currently do. And in many ways, it's really a matter of whether you want to be a scientist, whether something specifically inspires you. But once that's happened, the specific problems or the specific questions that you work on are really an accident of where you found yourself and what other people were doing around you and what got you interested. Um, I guess that's all I have to say.
Well, that's a very interesting story how you switched, kind of switched, from physics into the life sciences is really interesting. A story there, and it probably is a key to yeah, to being able to merge these fields, to have such, interest to develop such good work and such an interesting work. And then how did this project come about? Is there maybe a background story around that? Was that, something that developed based, on previous interests, or was it really hard, easy to get funding for, whatever the backstory is? It's really interesting to hear. Thank you.
So this particular project revolves around trying to understand how the mechanical properties of DNA regulate various biological processes. And in oftentimes in science or in biology or any branch of science, for example, um, we tend to think about science being driven by this flow chart of you have a hypothesis, then you test your hypothesis, then you refine your hypothesis, and the cycle continues. But we do every once in a while take a pathway that's not exactly as streamlined, and I guess that was something what went on with this project. Oftentimes, before you even have a hypothesis, you need to make a new observation about the world. And so a really good way to generate hypotheses is to develop techniques of making new types of measurements without any bias about what those measurements that you will make down the road would tell you about the world. And so that's really where this project came along. You see, for decades before this, people had measured mechanical and physical properties of DNA, but we developed this technology to make those measurements in very, very high throughput and to understand how those measurements would change with sequence. And because we had a tool, it allowed us to make many new observations. And so we went ahead and make those observations to see what kind of hypothesis that would come about from those observations. And we saw that based on these observations, we could come up with several ideas about how the physical properties of DNA might drive specific biological processes in general, might encode regulatory information. And that really served as the basis for this current paper that is being shared. And it was really a means of testing all the hypotheses that came about because we developed this tool in the first place to make novel types of measurements on DNA.
Yeah, that's really interesting, and I'm glad, the this project came about and got realized. And yeah, so everyone, the slides are pinned on top, and Ashkash, the stage is yours. Thank you.
Okay, thank you so much, Katerina. Quick question is that like, how does questions work? I mean, would people? Yeah, go ahead.
Yeah, so there are two ways of asking. One is raising the hand and coming here on stage, like Jeremy just did. The other one is using the chat, posting comments there and questions, and I will monitor it for you, so you don't have, you can just focus on answering and presenting. It's really up to you if you open up for questions in between, or if you rather would like people to wait until the end. Usually, most people just wait, but yeah,
Okay.
it's really up to you.
No, it's fine. I mean, I would like this to be as interactive as possible since it seems like a more kind of an informal way to present rather than formally with slides. So I'd be happy to like if you notice some important questions that are being raised, maybe you could point me to it, and then I could pause and talk a little bit about it and take that chance to catch up on questions. Yeah. Does that sound good?
Yes, perfect.
Okay.
Thank you.
All right. So let me start. Um, so So my lab is in Durham University. It's a medieval town in the north of England, and here's a picture of the iconic Durham Cathedral. And this is where we are studying DNA mechanics and trying to figure out how the physical properties of DNA impact various types of biological processes. So let me get straight to it. If you look at the complex functions that all cells need to carry out, it's just mind boggling. Cells have to, for example, transduce energy, they have to grow, they have to sense the environment they live in, they have to reproduce, they have to evolve, and there are many, many other tasks that cells have to carry out. And very broadly, proteins and other macromolecules carry out these various types of functions. And information teaching the cells how to produce these proteins and other macromolecules is stored in DNA. And this is, of course, very basic stuff. And I'm pitching this for a extremely broad and general audience. And chances are that a lot of you already know this. So feel free to zone out. Otherwise, stay with me. Um, so what is DNA and how does it code information that cells need to produce proteins and other macromolecules? So physically, and this is slide 3, physically, DNA is a very long string. It's a polymer. And if you zoom in on any one region of the polymer, as I have done on slide 4, and it's essentially two strands which are wrapping around each other. And along each strand is a sequence of up to 4 chemical bases appearing in any random order: A, T, G's, and C's. This is, I guess, part of general knowledge these days. And somehow, this sequence of bases along the DNA encodes information. And it's very much like how, for example, in slide 5, I have this little cartoon of how the sequence of characters can encode some meaning when encoded via the English language. And it's very similar. The sequence of bases along DNA can encode some information, which cells can decode via what is known as the genetic code and use that information to produce proteins. So now uh, I'm on slide 6 at this point. Just storing information in DNA is pretty useless unless you can do something useful with that information. For example, you have to copy that information and replicate the entire molecule of DNA when the cell divides, so you can pass it on equally to the two daughter cells. You need to repair this information in DNA when it gets damaged because of chemical factors, because of UV radiation or whatever. Then DNA, remember, is a very, very long string of bases, and it needs to be packaged properly within the crowded environment of the cell. And finally, the information needs to be transcribed, which is a fancy way of saying that it needs to be read. You just can't store the information. You still have to read it when you need to produce proteins to carry out the tasks that you need to carry out in order to continue life. So, how are these critical processes accomplished? Well, it turns out that there are specialized proteins within cells which bind to DNA at various regions and carry out these tasks of copying, repairing, packaging, or transcribing the information in DNA. Which brings us to the question in slide 7 for what exactly are these DNA binding proteins and what are they doing locally to DNA? Now, DNA binding proteins are proteins which bind to specific regions of the DNA, and then, based on their chemical properties, they can locally modify the DNA or repair it or read the sequence of bases along it and so on. But from our point of view, the most interesting and the key thing to bear in mind is that these are real physical attractions between DNA and the protein of interest. It's very much like you touching a long string somewhere. It's not just an interaction that you keep tab off on a computer or on a database. And so, whenever proteins bind to DNA, there is some kind of physical distortion or deformation to the DNA molecule that is being induced. Why? Because it's a physical object touching another physical object. And on slide 7, there are two examples from among millions of examples, not millions, thousands of examples of DNA protein interactions. In the first instance, that yellow cylinder is a protein core called the histone octamer. It's not important to go into the details. And the black string is DNA. And DNA wraps around this core, this cylinder, forming the structure called a nucleosome. And it is very much like how you would wrap a string around a spool in order to properly package it. In fact, that's one of the easiest ways in which you can organize and package a jumbled piece of string. It's by wrapping it around a spool. And nature does the same with DNA within the cell, except all of the DNA is not wrapped around just one spool, but there are many, many such spools throughout the genome, and each spool wraps about 2 turns of DNA, forming these structures called nucleosomes. But the important point is that it involves extensive DNA bending. As you can see, DNA bends and forms almost 2 circles as it wraps around the protein core. And on the right is another example of a molecular machine called a polymerase, which sits on DNA, walks along it, and reads the sequence of bases along its length. And in order to gain access to the sequence of bases, it needs to locally unwind the 2 strands. And so that's yet another example of a DNA protein interaction that requires mechanical distortions of DNA. Now, if we move to slide 8, what I'm trying to suggest here is that whenever objects undergo, if you try to mechanically deform an object, most objects resist deformations. If you press a spring, the spring pushes you back. If you try to bend a rod, well, you need to put in a lot of energy because the rod resists bending. And it's no different from DNA. If you try to bend DNA, if you try to twist DNA, DNA has mechanical properties that allow it to resist these types of deformations. And those mechanical properties have been very well characterized for over 30 years. And so, in many ways, the ease with which these DNA binding proteins can carry out the critical tasks that they need to carry out, this ease can be regulated and controlled by what the mechanical properties of DNA are. If DNA is very hard to bend, then maybe it's going to be very hard to form a structure like the nucleosome. If DNA is very hard to twist, it's probably going to be very hard to locally unwind the 2 strands of DNA and allow processes like transcription to take place. Then, so that apart, the next idea that has been gaining traction for quite some time is that not only does DNA have mechanical properties that allow it to resist deformations, but that those mechanical properties in turn depend on what the local sequence is, which means that throughout the length of the DNA string, its bendability or its twistability are not constant, but they vary depending on what the local sequence is. Which means that sequence can actually control and regulate DNA deforming processes such as nucleosome formation or transcription via the impact that it has on the local mechanical properties of DNA. And now, going back to the analogy of the English language, it's very much like how the same set of characters, PAIN, when encoded in English, convey a meaning, as shown in that cartoon in slide 9. But that same set of characters, when encoded in the French code, conveys an entirely different meaning. Likewise, in the case of DNA sequence, the sequence, when decoded via the genetic code, gives cells information on how to produce proteins. But there is a mechanical code as well, which also controls what the local mechanical properties of the DNA are, which in turn is going to control how easy it is to manipulate or to repair or package or copy the protein coding information that is there in that region of DNA. So, broadly, we want to decode this mechanical code. We want to understand how, throughout the entire genomes of organisms, variations in sequence lead to variations in the physical properties of DNA. And in turn, how does this control and regulate the thousands of DNA deforming biological processes that need to take place throughout the genome in order to properly carry on the functions of life? So that was sort of a very broad introduction, and this might be a good time to briefly pause for a few questions if there are any. Um,
Yeah. In case anyone has questions, please unmute or put it in the chat, and I'll read it out.
Um, Hi, Kat. Hi, Akash. So I think like
Hi.
I probably would like to sort of ask,
Yeah.
sort of based on what you're explaining so far, this is actually really fascinating, and really curious to see where what you to find out like what your findings to
Mhm.
essentially look into what your findings are. That being said, I want have like one question. I may have like other later on, but are there like, for example, transcriptions of proteins that make it impossible because the the mechanical deformation of the DNA is rather impossible. So just to explain, what I mean is that suppose that you have sequence of base pairs, right? Um, is there sort of like an RNA sequence that that makes it harder to bind to this DNA sequence with predefined base pair sequence because of the mechanical deformation, and vis-a-vis that would lead to um, such a protein not be synthesized. Does that make sense?
Yes, absolutely, it makes sense, and that's a great question. So I believe I'm in a broad sense. What you're asking is that can the expression levels of proteins be controlled based on what the mechanical properties of the underlying coding DNA are? And yes, that's very much the case, because in order to properly express proteins, several things need to happen, like polymerases needs to bind to DNA, nucleosomes need to form correctly, and each and every one of those important DNA protein interactions, and I'm going to show you examples of this, depend and can be controlled by what the local physical properties of DNA are. So there is this underlying mechanical regulatory code that is encoded in sequence, which controls how easily the genetic code can express itself or can transcribe itself. I'm not sure if that was enough, but I'm going to give you examples where uh, processes like transcription are where we think processes like transcription are impacted by DNA mechanics.
Yeah, I'll be really happy to learn about that. Yeah,
Okay.
but thank you.
All right. So let me continue for now, and we can get back to questions later. So so we're on slide 10 at this point. Now, and at this point, I'm kind of changing gears and jumping over into more technical stuff. All So if there are important questions or clarifications, please do mention it. So we had this idea that the local physical properties of DNA can depend on sequence, but is that really true? And here's what is known as a FRET, a fluorescence resonance energy transfer, which Katerina introduced earlier. So this is a FRET-based experiment that confirms this suggestion that mechanical properties do in fact depend on sequence. And this is not to say this is the only way people have shown this. People have shown this over many years using many different methods. But in this experiment, we have a short DNA fragment. It's about 100 base pairs in length, which is very short as compared to the length of the genome. And on the ends, there are what are known as complementary single stranded overhangs. That's just a technical term for Velcro. Basically, the ends, if they come close to each other, can stick. And we put in two fluorophores. These are molecules that absorb light at a certain wavelength and they emit light at a different wavelength. And when the fluorophores are far apart from each other, as is the case when the molecule is linear, when the ends are not in contact with each other, then there is a certain optical signature to that which we can detect. And then when the ends do come close to each other, there is a different optical signature which we can detect. And that way we're able to infer what the shape of the molecule is. Is it linear or is it looped? And the premise or the hypothesis going into this is that if this DNA fragment is very, very bendable, then this molecule should loop really quickly. And if it is rigid, then it should not be able to loop very quickly. And on the right are kinetic curves for percentage in the loop state as a function of time. And you don't have to go into the technical details. Every curve represents a different sequence. It suffices to say that when sequences change, we see that the probability of looping dramatically changes by more than the rate of looping changes by more than an order of magnitude. So the takeaway message is that DNA sequence can dramatically impact what the local bendability of DNA is, and perhaps other mechanical properties as well, like torsional rigidity, although we don't have a ready means of measuring those. So let's go back to this flowchart on slide 11. We're talking about processes that involve DNA deformations. Those processes are likely impacted by what the mechanical properties of DNA are, and we know that those mechanical properties of DNA in turn depend on sequence. So, this automatically leads to the big, broad question that we want to answer, which is that does sequence store regulatory information encoded via a sort of mechanical code, which can regulate diverse DNA deforming processes throughout the genome? So, what do we have to do if we want to answer that question? Well, it's simple. We have to take I'm on slide 12, by the way. We'll have to take the entire genome of an organism, chop it up as a sort of as a series of short DNA fragments, perform those FRET experiments I discussed about on each of those fragments, and use that data to create a map of how DNA bendability varies with position throughout the genome. And then we will compare that map to other known genomic data sets, which tell us, for example, in which regions of the genome are certain processes happening, in which regions of the genome are certain DNA deforming processes not happening. And we try to see if there are correlations. For example, is it really true that regions along the genome where DNA bending processes need to happen, are those regions, by virtue of their local sequence, also very flexible to accommodate such processes? Are regions where DNA bending processes should not happen for other, in order to serve other functions, are those regions also more rigid? And we can really begin to answer questions like these. Except the only problem is that we cannot do FRET experiments in such a large number of DNA fragments. It's simply impractical. It's too slow a method. So we developed a different method, and this is based on next generation sequencing, which is a very cutting edge modern technique of of uh, of sequencing a large number of DNA fragments. For example, if you give it like even up to 1 billion short fragments of DNA, each fragment having a different sequence, we are now able to tell exactly what those billions of sequences are. And this is a completely revolutionary technology that has changed the way we do molecular biology over the past 10 years or so. So here again, what we do is we start with a very large number of short DNA fragments, let us say, which span an entire region of the genome, pretty much like what I showed in slide 12. We just pool those fragments together and allow those fragments to loop as fast as they can for about a minute, and remove the molecules that have failed to loop, preserving only the ones that have looped. And then by sequencing, and we don't have to go into the technical details here, we can measure and quantify for every sequence that we originally had, how likely is it that it will end up in this looped pool, and how likely it is that it would not have been looped in a short amount of time. And that likelihood is what we would quantify as the bendability or the cyclizability of that sequence. So that's a sequencing based technique that we developed. And even if you didn't get the details properly, you could just imagine that we now have a method that allows us to measure how, with the variations in sequence throughout the genome, the local bendability or the cyclizability of DNA also varies across the entire genome. And then, as I said before, what we now want to do is compare these mechanical maps along the genome with other available genomic data, which tells us where DNA deforming processes need to happen, and see if there's a correlation. And we indeed see that. For example, if you look at slide 14, on the right, the two graphs, and the bottom is a graph of typically along a gene where nucleosomes are known to form. The y-axis is nucleosome occupancy, that's the blue curve. And right on top is a map of along those same regions how the bendability of DNA varies. And we were amazed to see this very good correlation. That is, regions where nucleosomes are avoided, such as if you just go upstream of zero around minus 150, you have a large dip in nucleosome occupancy in the blue curve. And that also spot on corresponds to an extremely rigid DNA region. And then wherever there are peaks in nucleosome occupancy, we also see peaks in local DNA bendability. And to me, this really suggests that there is sort of a benefit for evolution to have selected DNA sequence that makes DNA in some regions very rigid and makes DNA in other regions very flexible, because in turn that facilitates the formation of nucleosomes in such regions and prevents the formation of nucleosomes in other regions. And in turn, this of course serves downstream processes such as transcription. So, so okay, so we have made a measure of DNA bendability as a function of position, but can we now really decipher this mechanical code? If I give you a piece of DNA sequence, can you look at it and decode it and tell me what its mechanical properties should be? And that question is what we're trying to address in slide 15. We start with a large pool of DNA sequences. We measure their bendabilities based on this genomic technique that we have developed. And now we want to develop some kind of predictive models that sees the patterns and like figures it out and allows us to predict bendability from sequence without having to run these measurements over and over again every time. And that really was the crux of the current paper which we're discussing. So let me tell you a little bit about these sequence patterns that we have spotted, which seem to point hints to how this mechanical code is written or is encoded. And we're on slide 16, by the way. So based on our measurements, the first sequence feature that we decided to investigate as to whether it has an impact on DNA mechanics is, of course, the GC content or the AT content. That's the simplest feature that one can think about. And we saw that no, the way we measure bendability, something as simple as the overall GC content or the overall AT content is completely uncorrelated with DNA bendability. That's the first graph on slide 16. So we decided to make it a little more complicated. Instead of looking at just GC content, we looked at the contents of all possible 2 nucleotide pairs. Now remember, there are 4 nucleotides, A, T, G, and C. So there are a total of 4 times 4 or 16 possible pairs. And what the middle graph is quantifying is simply this, that in a given sequence, in a given 100 base pair sequence, if I have a lot of these pairs, does it make on average a contribution towards the bendability of that fragment? And at the extremes are CG and TA. So CG makes a very negative contribution to bendability, which means that sequences that have a lot of CGs in them generally tend to be rigid, but that's not to say that they are all rigid. And sequences that have TAs in them a lot generally tends to be more flexible. And it doesn't end there. We can quantify the contributions of all of them. And we did this by really detecting and quantifying and doing statistics on the data that we have obtained. And then the last figure is a little more complicated, but let me just explain what it generally means. It means that not only is the number of times these dinucleotides are present in a sequence matter, but how they're distributed along the sequence also has an important effect, in that certain pairs of dinucleotides, when they are separated at the helical repeat of DNA, they would make a positive contribution to bendability. And when separated at half the helical repeat, would make a negative contribution to bendability. And we again spotted these patterns by essentially looking at the data and trying to figure out what those patterns are computationally. And we put it together into a sort of predictive model, which is what slide 17 shows. And if you look at the graph on the right, remember in the bottom panel is a map of how the known positions of nucleosomes vary along genes in yeast. And in the middle panel is the predicted measure of DNA bendability, while the top panel is the actual measured values of DNA bendability. And the two red curves, the middle panel and the top panel, really recapitulate most of the broad features. And so that kind of tells us that this model we have come up with to decode the mechanical code of DNA does do a reasonable job in pulling out the important mechanical features that we think have important biological functions or consequences. Okay, so so that's at this point. Let me pause for one or two questions at this point. We're like 2/3 the way done, and then I'm going to show you some examples where we apply this mechanical code to different biological situations where we think DNA mechanics is important, and then I'll answer questions after that. But are there any questions right now?
I think Jeremy has a question. Please go ahead. Oh, yeah, and Perlook. Yeah, go ahead, both of you.
I got to go back to the. Excuse me. Let me get the
Yeah.
graph back up. Okay. On 15 is the middle graph. Does that apply to everything? Like, can I use that as a graph to say that bendability is expressed in because I love that graph.
I'm sorry, which slide number are you on?
Okay, it looks like it's 16 and
16 middle.
chart, the middle chart.
Yeah, I understood. Yes. So no, absolutely not is the answer to that question. Um, Here's what it really means. It means that if you give me, let's say, 100,000 random sequences, and for each sequence, I sort of quantify how many times each of these dinucleotides occur, and then I'll see that on average, if there are a lot of CGs in a sequence, they tend to be rigid. On average, if there are a lot of TAs in a sequence, they tend to be flexible. But that is, it's like we obtain the number on the y-axis is really the slope of a scatter plot of number of times the nucleotide occurs versus the bendability of the sequence in question, and that's just the slope that we are plotting here. there are millions of examples in which you have sequences with a lot of CGs which are extremely flexible. And so this is not something that you can just look at and read off bendability from. And not only that, let's look at some of the not so interesting dinucleotides like AA and AT, which are right in the middle, which don't seem to have any contribution at all. Well, it turns out that in a sequence, the overall content of nucleotides like AA doesn't really correlate with flexibility. But when there are two AAs separated by 10 base pairs, it tends to make the DNA very flexible. Whereas when you have two AAs separated by 5 base pairs, it tends to make the DNA rigid. And so it's not that overall content that's contributing, but the manner in which they're distributed along the DNA sequence. And we took all of that into account when we built the predictive model, which we subsequently used to make the predictions in slide 17. And even then, the model's not that good. I mean, if you look at the graph on the left in slide 17, the correlation coefficient is only 0.5 between predicted and measured values. And that's really a reflection of how complex the problem really is. So hope that gave you some idea about the question.
I appreciate it.
Yep. All right, let's go for it now. I'm going to continue, and we'll come back to the questions later. I'm almost done telling you whatever I wanted to say. So let's go to slide 18. So we have now a model that we built on the basis of the lots of measurements that we did, which allows us to predict how local DNA bendability varies with position along large regions of the genome. And broadly speaking, we used it to show that the way in which sequence modulates DNA bendability has played a functional role in ensuring that nucleosomes throughout the genome are organized in a manner that's functionally important. So, of course, in biology or in any science, we're always looking for general principles. So we're not content with having a certain principle work in a very, very specific and limited way. So we asked, okay, nucleosome organization around the start sites of genes seems to be impacted by DNA bendability. Is it true for nucleosomes organization around other genomic loci, which are not necessarily the start sites of genes? And we picked an example. There's a transcription factor called CTCF, which serves many purposes. And it seems that nucleosomes are also formed in very stable manners around CTCF binding sites. And that's shown by the plot below. We have a plot of nucleosome occupancy as a function from the center of the CTCF binding sites. And there are clear peaks, which telling you that here, you have a nucleosome here, you have a nucleosome here, you have a nucleosome here, and so on. And those peaks also corroborate with our predicted locations of peaks in DNA bendability as written in sequence, really suggesting that sequence encoded DNA mechanics might play a regulatory role in organizing nucleosomes, not just around start sites of genes, but around other genomic loci as well. But that was still about nucleosomes. What about processes that have absolutely nothing to do with nucleosomes? And let's skip slide 19 and go straight to slide 20. And here's an example of a situation where, of a biological situation where you have extensive DNA bending, but there is no nucleosome involved. So there are proteins, there are enzymes called topoisomerases, which twist DNA, serving many important downstream processes. And in the process of twisting DNA, they need to significantly bend the DNA molecule. And here's a specific topoisomerase I'm looking at called DNA gyrase, which has a specific site that it really likes to bind to along a genome of a phage. And what I did here on the graph, in the graph on the left, is predicted intrinsic cyclizability as a function of position along that entire phage genome, which is known to have just one gyrase binding site. And as you see, along that entire genome, that one sharp peak in flexibility, which is zoomed in on the right, corresponds spot on to the one known position that gyrase is supposed to bind to, really suggesting an impact of DNA mechanics in controlling a process that has nothing to do with nucleosomes and in a very, very different type of organism. So, and the last thing that I wanted to leave you with is that, and maybe some of you are already thinking about this, is that things written in sequence are not really cannot really be controlled that much by processes other than evolution, which takes many, many generations to work itself through. So do cells have some control over the mechanical properties of DNA, which they could do to, for example, respond to changes in environmental conditions, to respond to other stimuli that they may be subject to? And so we asked, for example, do any of the chemical alterations to DNA that take place during the life of a cell have any impact on DNA mechanics? And one of those chemical alterations is methylation. It's a process whereby a methyl group gets added onto cytosines, which precede a G. So if you have a C followed by a G, oftentimes the cytosine is methylated. And we asked whether this process of methylation would have an impact on DNA bendability. And we found that yes, indeed, it does. So on slide 21, in the black curve is DNA bendability or cyclizability as a function of distance from the start sites of genes. 0 is the start site of genes. That's the black curve. If you recall what we showed earlier, there is this sharp dip in bendability corresponding to a region where nucleosomes shouldn't form. And then there are peaks as you go into the gene body, which is what you see. And then the orange, actually, I don't know what color that is. The orange yellow is the hybrid curve, is the map that you get if you were to methylate the DNA. You sort of lose this contrast between the valley of the dip and the peak of the dip. That contrast becomes a lot more shallow. And that leads us to this very exciting possibility that if DNA mechanics can control critical downstream processes, then maybe biological programs like development or diseases like cancers, which can change the methylation landscape, could achieve a part of their downstream effects by altering those physical properties of DNA itself. And so with that, I just wanted to put up this one sentence in the conclusion slide. And let me just end on this broad note that if you look at the history of molecular biology, big strides have always been marked by stages in our understanding of the way in which DNA stores information. The early big event was our discovery that DNA stores protein coding information in sequence via the genetic code. And that in many ways ushered the modern era of molecular biology. But then eventually we found out that there are other ways in which DNA stores information. For example, we found out that DNA stores information not only in the sequence of bases, but also in the manner in which these bases are chemically modified, what is known as epigenetic modifications. And that ushered in an entirely new and vibrant line of investigation. And then more recently, we found out that no, there are yet other ways in which DNA sequence stores information. DNA stores information, for example, in the overall three-dimensional shape of the genome. And that is a very active area of investigation. And what we are suggesting here is that even beyond all of that, DNA sequence also stores information in the manner in which sequence modulates the mechanical properties of DNA. And so there are possibly in biology, there are a myriad of different codes, each using the sequence of alphabets along DNA in order to encode a different type of meaning, just as there are different languages which are the equivalent of different types of code using a similar set of alphabets in order to encode different meaning or serve different functions. So in slide 23, I'd like to acknowledge the people who are involved in this work, who are still involved in the work, have been involved in the work in the past. So my lab at Durham is very new. So I have two students, Aditi and Bailey, who have been working on taking this work further and developing a lot of the technological basis for some of the future experiments that we want to try. And then at Johns Hopkins, this work was, of course, done in collaboration with Johns Hopkins, specifically Tekji Pa's group and members of his lab. And we really were instrumental in developing the early assays and the methods to measure DNA mechanics and the funding. And once again, thank you very much for having me here. I was very interested, and it's been great. I've been looking forward to this, and I hope now I can take questions and
Yes.
answer anything you want to ask. Yes, go
Yeah,
ahead.
so I'm very intrigued with this in terms of early stages of epigenetics in terms of embryology, for instance. Is there any linking of this as being able to transmit perhaps an initial naturally occurring mechanical gradient from the collections of cells that would transmit through the cell membrane such that it would affect the DNA, kind of leading to the first generation of expression of morphogens, and then even ongoing as well in terms of once tissues start to form, substrate adhesion molecules are made on the surface, again transmitting the mechanical properties of the tissue formation and migration through the cell membrane to again can make more types of morphogens that can be used in the further development of embryology.
Absolutely. I probably didn't catch every detail of the question, but I can tell you what I've thought out about how mechanical information can be transmitted from the environment to within the genome. There's a well-studied linkage that links the cell wall. There's the actin cytoskeleton of the cell, which is in contact with the nucleus. In turn, the nuclear membrane is in contact with DNA through laminin-associated domains and various other proteins that are involved in establishing these contacts. And so one can certainly imagine that if you pull and push on cells, or if they find themselves in different mechanical situations, that information can make its way to the outer surface of the genome, which is in contact with the nuclear membrane. And then really understanding how that would propagate deep within the genome and impact local transcription throughout the genome is honestly a question that requires us to deeply understand the mechanical properties of DNA itself on various scales.
Mhm.
Like if you
Yes.
pull a piece of string, how it responds a large distance away from where you gave the initial tug, it depends in very complex ways on the mechanical characteristics of the string. And so, if as we continue to develop more, as we continue to make more measurements of the types of mechanical properties and how they depend on sequence, we would hopefully find ourselves in a position to be able to talk about how long distance mechanical signals can propagate through DNA.
So fascinating talk. I've spent more time, worrying about protein structure but I've always been curious, and RNA structure is quite fascinating. But the tertiary and quaternary structure of the chromosomes has always been a topic of interest. This work seems foundational for models of predicting that tertiary or quaternary structure and how that might relate to exposed sites that would be more kinetically available. Could you comment more on that? And perhaps as a follow-on question, are you involved or aware of work, building on these models to either, through empirical calculations or, AI type AlphaFold sort of efforts, and to, work towards a better means of predicting the tertiary and quaternary structures?
Great question. So we have concerned ourselves very much with, I guess, what you would call primary structure rather than tertiary or quaternary, because look at the scale at which we are looking at the process. We're looking at the process on a 100 base pair length scale. And that is, of course, a very important scale in biology because it is the sort of length scales at which proteins interact with DNA. So, which is why we decided to investigate there. Now, I guess your question is, to what extent do these very local measurements of DNA mechanics affect the large scale structure and architecture of the genome? And there is no easy way to answer that question. We must bear in mind that if you grow bigger and bigger in scale, you're also going to average out the sequence dependent effects. So one has to be wary about how much would sequence dependent variations in mechanics at smaller length scale manifest themselves when you talk about larger length scales. But of course, when it comes to RNA or proteins, we know it does. The way the quaternary or the tertiary structure forms very much depends on local interactions. But whether it's the case with DNA or not, or to what extent, is of course something that we have to look into. But I do want to talk a little bit about how a large scale structure of the genome could have an impact on local states and what relation DNA mechanics might play in it. And this is really something that we are currently investigating in my lab. You know, in bacteria or in like E. coli or even cyanobacteria, we know that the superhelical density of the genome is this overall architectural parameter that is responsible for establishing the overall degree of compaction of the entire genome. So you could sort of think about it as a parameter that directly affects a large scale tertiary structure of the genome. And that superhelical density of global superhelical density of the genome also somehow controls the transcription, the expression levels of hundreds of genes scattered throughout the genome in a very complex and very specific manner. It knows that it affects this gene, this gene, this gene, and not this one, and this one. And it does so, and like how this tertiary structural parameter controls local transcription is broadly unknown. And we have a hypothesis that perhaps the local bendability or the torsional rigidity of DNA can respond to global supercoiling levels and accordingly determine what the local twist of the DNA is, or how much compact plectineme structures are forming locally, which in turn would impact transcription. So to sum up, we are investigating situations where global structure and tertiary structure could impact local processes, and that this impact, the manner in which it impacts it, certainly is directly related to the way in which sequence controls DNA mechanics on a local scale.
So thank you. I was also fascinated how you looked at the methylation of the DNA and how that affects, and just to step way out in conjecture land about tertiary structure. Um, in one sense, as we age, the methylation increases in some cases, and I've always been curious if that has an impact on the exposure of certain regions of DNA, and that is there, some type of progression of exposure through methylation um, making some genes more available or less available with time. But it does seem like even just down to the local mechanical properties of whether the sequence is methylated or not may at least play into a progression of exposures for different genes. Any thoughts on that?
Yeah, absolutely. So if you look at on average what we find that on average, the more methylation you have, the DNA sort of tends to become more rigid. Although this is average, how it behaves by way of rigidity or flexibility depends on what the local sequence is. For example, if you have a CG being methylated, but it is surrounded by a very specific sequence feature, then that methylation could, make the DNA more flexible. But on average, it tends to make it rigid. If DNA is locally more rigid, one could argue that it would increase its local uh, exposure, but at the same time, it would make it hard for binding proteins to deform the DNA molecule, which is something what most binding proteins would want to do anyway. So there's a trade-off to that, and like what you mentioned, that during various progressions, be it aging, be it development, or be it during the progression of a disease, the methylation landscape changes, which has important downstream consequences for transcription, for example. And some of that can certainly be explained because methylation serves as like a landing pad which recruits various factors which do many things. So some of that can certainly be explained. But what our findings suggest is that maybe we also need to think about how methylation changes downstream processes because it alters the local flexibility or the exposure of DNA. And we're really at the starting steps of trying to do that. We don't even know fully well how the methylated landscape behaves flexibility wise, and we are sort of characterizing that right now.
Thank you. Fascinating work. I'll pass the mic.
Thanks.
Yeah, go ahead, Abyss, and then Dr. Shah.
Oh, since I already asked, I did ask a question like early on during the presentation, so I will yield to Dr. Shaw first, and then I can go after her.
No problem. Thank you so much, Elvis, and thank you so much, Akash, for your wonderful presentation. And my question from you based upon we know about the epigenome and how it can impact the changes. And you mentioned briefly about the cancer. And I was just wondering if we want to think about your research and the use of your research, for example, in the case of the tumor heterogeneity and how it can epigenetic can impact it. So do you have further information? However, we can think about the aging and reversing of the aging, which it can apply for the cancer as well. So I was just wondering any further information that you can share with us.
No, absolutely. I mean, cancer and aging are not separate. Cancer is a consequence of aging, and in many ways, cancer is a disease of aging. So we could look upon them in the same way. Now, we know that methylation changes as you age. There are some very interesting works done on what is known as the methylation clock, which is like this pattern of methylation which you can detect and very, very accurately use to quantify the age of the cell or the tissue. Whether it's a cause or a consequence of aging is unknown, so one has to be careful. If you talk about reversing, it's not a simple manner of just reversing the methylation profile and reversing the course of a disease or the course of aging. It could very well be a cause, it could very well be a consequence, or like most things, it could be a bit of both. And we don't know that. But all all I am suggesting, or what we could think of in the light of the work that we have been doing in our lab, is that when we look upon the effect that methylation has, we need to consider the effect it has on the physical properties of DNA and chromatin as well, because any other form of effective methylation would be very, very specific. It's going to affect specific CpG recognition factors, or it would affect proteins that specifically have those binding factors. But if it can affect more general characteristics of the genome, like its architecture, its shape, its accessibility, and its local flexibility, then that's one way in which it can have a much more wider, broader impact on downstream processes. So it's a bit like alcohol, which just goes and affects everything it can, more than, and it's not very specific at all. So methylation, so mechanical changes as a result of methylation changes which occur during the progression of a disease could very well alter that progression, or could very well cause the downstream deleterious effects because it changes a large scale structural parameter of the genome.
And what about the, for example, a structure like zDNA and impact of them? How you just evaluate that one?
Yeah, I mean, I have not looked at zDNA particularly, and we know zDNA forms. We know it forms in the lab, of course. It's easy to form, but uh, uh, we're not exactly. I mean, as far as my understanding is that we're not too sure whether zDNA forms that often in vivo or not. But just zooming out a little bit from zDNA, if you just look at other sort of structures of DNA, like G quadruplexes, or or things of a similar nature, I honestly don't see a relevance of bendability in those cases. And it's easy to get carried away and think that everything can be related to DNA bendability, and it really cannot. That's all I can comment on. I mean, in the specific case of zDNA, I don't know. I have looked at G quadruplexes because lots of people ask us this question, and we didn't find any kind of relation whatsoever between DNA mechanics and G quadruplex structure formation. Now, there are other structural transitions that DNA go through, like the B to Z transition, like you're talking about, or the B to S transition, and whether DNA or melting transition, and whether DNA mechanics on the local scales would have something to do with those transitions, or something to do with the critical coefficients associated with those transitions. That's an interesting question, but I don't have the answer to that.
Thank you so much. I'm passing the mic to the next person.
Hey, Akash. Yeah, really good presentation again.
Thank you.
So I guess my question is that so I don't know if you've done work on this, but even if it's not, even if it's not, like if it's, I'm just curious, like if it's something that you're looking into, which is, do you think that there is some kind of mechanical strain, like for example, in single cell organisms that tend to create like repeats of sort of like interspace short palindromic repeats, like CRISPR, for example? And do you think that would create some kind of mechanical strain because of the sheer repeatability of certain sequences of genes in order to prevent, like, viral infection? And with that, I'm also curious what you think about like some portions of the DNA that do not go through the transcription process, like for example telomeres, if that's actually more of the mechanically enforced as opposed to like other factors.
Okay, so let's talk about the first point that you raised, which is, does repeatability of sequence features um, impose a mechanical strain to DNA? Is that what you were asking?
Right.
Right.
So yeah, exactly.
Yes. So okay. So let me tell you what we have observed so far, or we can just start with the simple example. For example, if there's a short sequence feature that tends to bend DNA in a certain direction, then if you have that sequence feature repeat every helical repeat, like every 10 base pairs, then those bends would get reinforced, would add in phase, and you would have an overall curved molecule. Whereas if those features were to repeat themselves every 5 base pairs at half the helical repeat, then they will cancel out and you'd have a more straight molecule on your hands. And we routinely see this when we analyze our bendability data. Now, you see, I didn't talk about it, but in our data, we can't really distinguish between whether a molecule is flexible in a dynamic sense or whether it is statically curved so that the ends are already close to each other, allowing that looping to happen quickly. We can't really distinguish between these two situations. And that's not a huge impediment because in biology, if it's statically curved, it would also facilitate forming bent DNA protein structures, which is really what we are trying to predict here. And we routinely find in our observations that there are sequence motifs like AAs and ATs, which, when repeated every 10 base pairs, tends to make DNA more statically curved. When repeated 5 base pairs, seems to have the opposite effect. And then we looked at certain regions of the C. elegans genome, which is it's known as the omega 4 region, where there is what is known as hyperperiodic DNA. Certain sequence features have a very strong periodicity as compared to other regions. And using our predictive models, we saw that those regions are also very, very flexible. And if we were to scramble the periodicity by keeping the sequence contents, the contents of A, T, G, and C the same, but if we scramble the sequence in those regions, we lose that enhanced mechanical flexibility or curvature. And that's yet another way in which hyperperiodic repeats of certain motifs that confer structural properties can certainly reinforce each other and create overall large scale curved or straight structures when repeated many times. Does that somewhat answer the question?
It does. I guess
Mhm.
my second question was on
Yeah.
why, if like parts of the parts of the DNA that do not regularly go through transcription, do you kind of
Oh,
account for
yeah.
that sort of mechanical effect to that?
Yeah, I haven't done studies, but it gets raised all the time, and it's super interesting. I think it'll be very interesting to look at telomeres. Uh, for example, and mainly from the point of view that even though it's not being transcribed, but the telomeres are abundantly bound with so many proteins and so many factors, and its degree of compaction or degree of exposure has important consequences for biology. So it would be super cool to measure mechanical properties around telomeric regions and see how it correlates, or whether we could make some sense out of it in the light of the processes that take place there. But I haven't done any such measurements yet.
Yeah, thank you so much. I'll pass the
Hmm.
mic to the next person.
Yeah, Jeremy, Philip, Amir, please go ahead and ask your questions.
If Amir, do you want to go?
No, go ahead, please. Thanks.
Thank you. Unfortunately, I couldn't hear the questions that Brother Abyss was saying, but thank you, Akash, for being clear in your response. Thank you again. Oh, let me start properly. Thank you for sharing your studies. Very inspiring. I'm an amateur in this. I'm a beekeeper, Sankofa scientist in that sense. So through observation and trying to understand my livestock, it has brought me into these type of realms where I'm interested in these. It's brought me into these conversations. It sounds like when you're talking about the silence language, a secret code and whatnot, similar to certs. Yeah, is it the certs? Or are you referring? Is this part to do with the certs type of thing, like certs 1, 2, 3, and 4, etc.?
Not really. Not really. Let me clarify. I mean, there is no there, there is no like special code here. It's not a clear cut code like the genetic code is. I mean, we know that sequence features impact mechanical properties, and in that way it's a code. But if you look at the genetic code, a certain codon always codes for this amino acids, no matter what you do to it, no matter what conditions you put it in. But the mechanical code need not be so specific. For example, under certain conditions, a certain sequence can code for a certain mechanical property. If you change the salt, if you change the osmotic stress, if you change the temperature, if you change the organism, if you change the epigenetic marks, it could be different. So we have to embrace the fact that this code is not as hardwired or as rigid as the genetic code is. And
Oh, most
it,
definitely.
yeah, and in relation to Circ proteins, I mean, I've never, I mean, I don't, I can't really comment anymore in that. Uh, could you explain to me why you felt that the similarity
Well,
is
yes,
there?
well, just the way
Yeah.
how you was referring them, like the mechanisms, and I'm looking at
Mm-hmm.
your slides,
Mm-hmm.
and in relation with like the where you refer to the twisting or the bending and
Yeah.
and the unwinding, the unbending, or the
Yeah.
reversal of that again sounds similar to the behaviors that the certs influence or relay or regulate in in that sense. And funny enough, I agree. If we're talking about bendability, and I think
Yeah.
bendability is the same as plasticity. Is bendability the
Mm-hmm.
same as plasticity?
You could think about it. Bendability is the ease with which you can bend, specifically bend, but so yeah, I'm sure they're
Okay.
related. Yeah.
Okay. But I
Mm.
understand what you mean by the
Mm-hmm.
bendability and then the rigidity of something.
Correct.
Hence, so funny enough, there
Yeah.
was a question so there's a lot of things I wanted to say, but there was initially the first question I kind of responded to was when you
Yeah.
mentioned to the colleague who mentioned Jeremy mentioned something, and you was responding to him, and you mentioned it was regarding slide 16 or 17, and you mentioned like a ratio of a number. You mentioned the last number was 5, and I think you said 3, but you could have said 8, but I don't know. But then when you was talking to Abyss, you mentioned it again. But guess what? I only heard the latter part.
Oh,
So it was either 5 and 3 or 5 and 8. Can you remember saying that number or
I
ratio?
remember saying 5.
Yes. Okay. Okay.
But what I meant when I said 5 was that like 5 is half the helical repeat.
Yes.
So uh, so DNA is a double helix, so the two strands are wrapping around each other as you
Yes.
go
And
along
there's
the
a 5
helix.
and 3 ratio. Yeah, between that in it. There's a 5 and 3 ratio or algorithm. Is that correct?
No, like
Oh,
every 10 base pairs, the
okay.
helix fully twists by one turn. What
Uh-huh.
that means is that if, let's say, you're sitting on a particular base pair and you're looking in one direction and you
Hmm.
see something, and then if you move over 10 base pairs and if you sit on the 10th base pair and look in the same direction, you'll still see the same thing because
Yes.
you've gone one full turn around. Whereas if you did 5 base pairs, you'd be looking at exactly like if you were looking towards the center of the DNA circle, you'd now be looking towards the outside because you're just half the turn
Yes.
away.
Yes.
So yeah, and so in that sense, what I meant to say is that let us say a certain very short sequence feature bends DNA in one direction. Now, if that feature repeats itself every 10 base pairs, those bend directions are going to add up. It's like, if you've played with circular train tracks that kids can play with, they fit into each other, and it's like you have one circle.
Yeah.
in
Yeah.
kids'
I
toys
fully
of train
understand. Yeah, I
train
understand what you
tracks.
mean.
Yeah,
Yes, I understand.
if you fit the tracks in the same phase, they'll make a larger circle. But if you fit them every 5, like if you fit one curving this way, the next one curving the other way, and the next one curving the other way, you'll essentially make a straight line, although it'll be wiggly up and down. And that's what's going to happen if you, for example, repeat the features that make DNA curved every 5 base pairs as opposed to every 10 base pairs. And we really see that in our measurements.
Yeah. So basically, yes, it was to do with spacing, and basically,
Correct.
yes, it was
Correct.
to do with spacing, and this. Well, obviously, sequencing obviously influences spacing. Spacing is part of its vocabulary, I'm assuming, or
Right.
you know, in
Absolutely.
that
It's
sense.
part of
Yeah.
the mechanical vocabulary. You can think about it
Thank
that
you.
way.
Thank you. So yeah, anyway, so yeah, I just believe that when I was listening to you, it sounded like how the behavior, the mechanics, the mechanics of how the
Yeah.
sirtuins, and obviously
Yeah.
I've associated the sirtuins to the
Yes.
royal jelly, to
Yes.
understand the royal
Yes.
jelly and the
Yes.
how it influences the queen and
Yes.
distinguishes as a larvae to have its
There
stem
was a very
potential
interesting paper
realized.
recently, right, on if you feed royal jelly, you can determine which bee is going to become the queen. I mean,
Yes,
I
very
only
true.
very vaguely
Yes,
knew about
that's very
it,
true.
but yeah, no, absolutely. And since you talk about similarities, that's a general theme in science. Like if you think about it, if you look at the macroscopic world, and I give you, let's say, a cable. You can bend it, you can twist it, you can feel these things. And here I'm talking about bending and twisting DNA, which is like as far removed from the world of our perception as you can possibly be. And we're quantifying its mechanical properties in exactly the same way as if we were talking about a cable that we can hold in our hands. That is amazing and fascinating to me. And I'm not. It's great that you see those similarities in contexts like sirtuins, which are very far removed from DNA, but that really reflects the fact that a lot of fundamental principles in science are applicable on a large diversity of scales, whether
What,
it is or a large diversity of systems. That's what makes these things really general and really interesting.
may I ask, why do you say that sirtuins is far away from? Did you say biology? No, DNA.
No, not scales, but systems as a
No, yeah, but
yeah.
did I hear you? Did you say that sirtuins is far away from DNA, or what did you mean? Can you repeat what?
Oh, all I meant in this context is that it is not it's not it's a different system. We're talking
Yes, of
about
course,
it's a different system, and the
of course.
same principles are and
To this
the
proteins,
phenomena, the sort of phenomena that happens in one system tends to happen in other systems as well. That's what makes
yes,
these things very interesting. It's
right,
more
it's
general rather than being specific.
true. Thank you. Oh, sorry for taking the time,
Mhm.
guys, and
No
I hope
worries.
that me
Yes.
being the fool allowed others to hear a bit more information. Whatever. Sorry. Thank you, guys. Peace and
No.
love.
Well, thank you for that. Please don't apologize for questions. Amir, did you have a question?
Yes, yeah, finally, it's my turn. Hi, everybody. Yeah, thank you, Akash, for the nice presentation and the excellent work. So I had a question. I was wondering if you checked and if you observed any correlation between the codon usage and the bendability of the DNA. So if you saw a correlation like in a region of the genome that more bendability is required, do the codon usage is the codon usage adapted to that need, or did you check at all? Thank you.
All right. Wow. I just say wow. I am super impressed that you asked a question, which is exactly what I'm working on right now. It just, I mean, I'm very happy. It means that I'm working on something that other people think is also relevant. And it also means that you've really grasped and you've thought deeply about what I was saying. Yes, absolutely. We do see that codon usage has a very interesting relationship with DNA flexibility. So we were asking this general question that, okay, a specific DNA sequence has a specific mechanical property, there's nothing you can do about it. But if you want to code a certain protein, you have a large choice of what the coding DNA sequence would be because of the degeneracy of the genetic code, which in turn is also, it's not, you don't have access to the full degeneracy, of course, because the codon usage limits it. but you still have a large number of sequences to choose from. And so the general question that I was asking is, to what extent is our need to is the need for evolution to select a specific mechanical property of DNA in a certain region of the genome, to what extent is it constraining the sort of amino acid sequence that region can encode for? Or, in other words, or does the degeneracy of the genetic code allow evolution complete independent control over mechanical properties in amino acid sequence? And so in yeast, what we did was this: we had measured DNA mechanics around transcription start sites, and we saw that it has a very clear pattern. DNA is rigid upstream of the start sites. Downstream, it goes through peaks in the DNA bendability goes through peaks, which correspond to nucleosome locations. And then we randomize codons along the gene bodies, taking into account the codon bias of yeast. And we found that those mechanical signatures are no longer there anymore. They're gone. And what it told us, and I think this is very interesting, is that the choice of codons that are there natively in an organism has been fine-tuned by evolution to best optimize mechanical properties along DNA which serve other important functions. And we're really trying to grasp even the question that we're asking right now, as to what extent does codon usage and codon degeneracy and the TRN and the codon bias link the genome's requirement and to the proteome's constraints that the proteome is constrained under. So, yeah, thanks for asking a very interesting question.
Thank you
Yeah,
for the answer. I have another question, if it's okay.
Go ahead.
Yeah, sorry, maybe you already mentioned it, but I missed it. So I was wondering how exactly the calculation is implemented. Is it a neural net or any other method?
Oh, you mean the calculation for predicting DNA mechanics?
Exactly.
Oh, yeah, we did
Yeah,
it
yeah.
two ways. So we made a large number of measurements of sequence and mechanical properties. One was a neural net, which is a black box type thing, and the other is we tried to pick out these short sequence features like contents of dinucleotides, the spacing between dinucleotides, and we put it together in sort of a regression model, a linear regression model, which gives us more physical insight. And they both perform reasonably well or reasonably poorly, no matter what you want to consider. Which really means that the code is very complex. The space of 100 base pair DNA sequence is larger than the number of atoms in the universe, and we are only sampling like 10,000 or 100,000 such sequences to us, which is an astronomical number made possible by next-gen DNA sequencing. If you compare it, earlier people could do it only for one or two or five different DNA sequences, and now we can suddenly do it for 100,000. But it's still tiny, and it's no surprise, therefore, that a training set of 100,000 is not enough to capture the full space, and we need to continuously rely on actual measurements in order to truly understand DNA mechanics.
Great. Thank you very much.
Thanks.
Yeah, thank you so much for the question. I have a last question if you
Go
still
ahead.
have time.
Yeah.
We had a guest speaker here. I don't know if you read the paper about using immuno-oligostorm imaging combined with visualization of the DNA histones to also look at the folding the folding of the genes with nucleosome resolution, and they found that during a different, like in stem cells, for example, versus mature cells, that the folding is different in order to so what they saw is that different genes are close together at some point and then not anymore at another stage of the cell. So based on your model, what do you think, what changes basically this folding, this dynamic folding, because you kind of predict that the DNA code itself changes the folding formation, but then the genes are used differently and folded differently um, throughout steps of maturation. So do you think it's methylation and so on, like, or what are you planning on expanding your model for those levels of complexity too?
Yeah, thanks for the question. It's a complex question. There's no doubt about it. It's not something that we can simply look at some simple measurements of DNA mechanics and comment. Like how we know, of course, that as development takes place, the three-dimensional folding of the genome changes dramatically. Genes that were like high C maps, for example, tell us that genes that were close together become far apart. Other regions of the chromosome are brought close together so that they can be co-transcribed, co-transcribed in the same transcriptional factory, and so on. They can share transcription factors, and then genes become far apart. There, we don't know. We don't know to what extent DNA mechanics on the local scale, which is relevant for a single DNA protein interaction, can manifest itself or can have any impact, if it does, on large scale rearrangement and folding of the genome. But this, I think, ties in to one of the earlier questions to which I was responding, which is how, let's say, external factors can influence the shape and the transcriptional profile throughout the genome. Um, uh, Methylation, I mean, I understand why you're talking about methylation, because during the course of development, that's the only thing that can change. Sequence is not changing. So if methylation controls DNA mechanics, it changes, and perhaps that induces some change in local folding, but it's in absence of further measurements, we can't really suppose that it has an effect that goes beyond the 100 base pair length scale that we have measured it. So I guess one has to be careful there and not get carried away too much. Other than methylation, there could be other factors as well which change DNA mechanics, which is not sequence, so it's not something written in stone. For example, different types of methylation. Different types of epigenetic modifications. It could be different DNA damage scenarios that could come about, or it could be chromatinization of DNA itself could change mechanical properties, like how the DNA's mechanical property and the mechanical property of DNA bound to protein, that structure can be very different. And as you go up and up in scale, those structures will more start to becoming more important. But really, all of that discussion was to say that the correct answer to your question is I don't know.
Yeah, thank you so much. I think we've been going on. I'm curious to see, what the future
Yeah.
brings in order to answer all those questions. It's
Mhm.
always like the best type of work that those, there will be a lot more questions. That opens up a lot more questions. Actually, that's the interesting one. So,
Yeah, absolutely. Yes.
yeah, exactly. And yeah, congratulations on this amazing work. And we wish you all the best for your future. And yeah, maybe one day when you, yeah, you could maybe update us in a year or so what results you came, to
Um,
discover. So thank you so
uh,
much.
Absolutely, and thank you so much for having me here. This was very interesting, and the questions are very, very insightful, I have to say. And it was very engaging, and I really enjoyed it. So thank you, everyone,
Well, thank
for
you.
having me and for inviting
Yeah,
me.
that's always the best when the speaker also enjoys it. That's the best
Mhm.
for us too. We are very happy to hear that, and yeah, I hope I hear you all back again soon. Thanks for asking amazing questions. It always so much more interesting when there are many different people asking questions. It makes the discussion way more interesting. And our next room is with Dr. Rai, who invented a new technology for precision engineering of proteins. So yeah, I hope to hear you all back soon next week. And thank you, Ashkash, so much for going through the trouble to come here to Clubhouse to talk with us. Thank you.
Thank you.
Okay, happy weekend, everyone. I'll close the room in 3, 2, 1. Bye, everyone. Thank you.
Bye, everyone.